The big question that can humans create a living cell? This video explores the frontier of synthetic biology technology. Imagine being able to design and build a living cell from scratch, a tiny, energy-producing powerhouse that can adapt to new environments, much like the first cell divisions that sparked life on Earth 3.5 billion years ago. For millions of years, evolution has crafted humans into complex beings, intricate machines composed of countless cells working in harmony. But now, armed with our advanced intellect and cutting-edge engineering, we're on the brink of replicating nature's most fundamental building blocks. Scientists today are making strides in synthetic biology, a field that blends biology and engineering to create lifelike systems. The question isn't just whether we can mimic a single cell's functions, but whether we can design something that not only survives but thrives, even if only for a brief moment. This breakthrough could revolutionize medicine, environmental science, and our understanding of life itself. Join us as we delve into the fascinating journey of creating life in the lab and what it means for our future. So creating a living organism from scratch is one of the most ambitious and intriguing goals in science today. While we've made significant strides in understanding how life began and in engineering biological systems, fully synthesizing a living cell purely from chemical components remains a complex and ongoing challenge. Let's break down where we stand and what's possible so far. First is understanding the origin of life, the fundamental question. Life on Earth is believed to have started around 3.5 to 4 billion years ago from simple organic molecules. These molecules gradually organized into the first cells through a series of chemical reactions. This process, known as abiogenesis, involves the formation of self-replicating molecules, the creation of cellular membranes, and the development of basic metabolic pathways. Next is the synthetic biology and minimal cells discussion. So synthetic biology is a field that merges biology and engineering to design and construct new biological parts, devices, and systems. One of the key areas in synthetic biology is the creation of minimal cells, cells stripped down to just the essential components needed for life. In 2016, scientists at the J. Craig Venter Institute, JCVI, created a synthetic bacterial cell called JCVI Syn 3.0. This minimal cell contains only 473 genes, the smallest number needed for a cell to sustain itself and reproduce. However, it's important to note that JCVI Syn 3.0 was made by transplanting a minimized genome into an existing bacterial cell, rather than building a cell entirely from scratch. Building cells from scratch is an enormously complicated process. Creating a living cell entirely from non-living chemical components is still a major challenge, but researchers are making progress in several areas. Scientists are developing protocells, which are simple, cell-like structures that can mimic some functions of living cells, such as compartmentalization, having a boundary like a cell membrane, and basic metabolic activities. These protocells typically consist of lipid membranes encapsulating a set of molecules that can perform basic chemical reactions. One of the key features of life is the ability to self-replicate. Researchers are exploring ways to design molecules and structures that can replicate themselves under certain conditions. While there have been advances in creating self-replicating systems, achieving the complexity and efficiency of natural cellular replication is still a work in progress. Some scientists are working on more complex artificial cells that can perform a wider range of functions, such as protein synthesis, energy production, and responding to environmental changes. These artificial cells aim to integrate various biological components into a cohesive system capable of growth and division. There are many challenges in creating living cells, but steady progress is ongoing. First is the complexity of cellular processes, an essential key to overall progress. Living cells carry out a vast array of interconnected biochemical processes. Next is replicating this intricate network in a synthetic cell is a significant hurdle. Energy requirements is an essential factor in building new self-replicating cells. Cells need a constant supply of energy to maintain their functions and power processes like protein synthesis and cell division. Also, designing an efficient energy system for synthetic cells is challenging. Genetic information is the key in building new life, another essential factor in creating self-replicating cells. Encoding and maintaining genetic information in a synthetic cell, ensuring accurate replication and expression of genes, is another major challenge. Environmental stability is another essential factor. Nature-made living cells are highly adaptable to their environments. Creating synthetic cells that can survive and function in varying conditions requires sophisticated design and engineering. Current progress and future directions are promising. While we haven't yet created a fully synthetic living cell from scratch, progress in synthetic biology continues to push the boundaries. First task is improving protocells to perform more complex tasks and self-replicate more efficiently. Next is incorporating more biological molecules and systems into artificial cells to increase their functionality and resemblance to natural cells. Further studying minimal cells to understand the essential components and processes required for life, which can inform the design of synthetic cells. This topic brings us to the role of CRISPR, clustered regularly interspaced short palindromic repeats, in advancing synthetic cells. Another groundbreaking technology that has propelled synthetic biology forward is CRISPR-Cas9, a revolutionary gene editing tool that allows scientists to make precise, targeted changes to the DNA of living organisms. CRISPR's ability to accurately modify genetic sequences has been instrumental in projects like JCVI-SYN 3.0, the minimal synthetic cell developed by the J. Craig Venter Institute. By using CRISPR, researchers were able to systematically edit and streamline the genome of JCVI-SYN 3.0, removing non-essential genes to create a cell with just the minimal set required for life. This precise editing not only enhances our understanding of the fundamental components necessary for cellular function but also paves the way for designing synthetic cells with customized capabilities. CRISPR's versatility and efficiency make it an indispensable tool in the quest to create living cells from scratch, enabling scientists to explore the boundaries of life and develop innovative applications in medicine, environmental science, and biotechnology. Conclusion: creating a living organism from scratch remains one of the most challenging and fascinating endeavors in science. While we've made significant progress in understanding the building blocks of life and engineering minimal cells, the creation of a fully synthetic cell that can autonomously grow, replicate, and evolve is still a goal for the future. Ongoing research in synthetic biology, protocell development, and our understanding of the origins of life continues to bring us closer to this remarkable achievement. Stay curious and keep exploring the wonders of science. If you found this helpful, give it a thumbs up and subscribe for more insights into the amazing world of biology and beyond.